everyone. I'm Lori. For those of you who weren't able to attend, we did have a session on Wednesday, and our wonderful support, Megan, has put that link in the chat if you want to go back and watch the recording. So let's start with letting you know that tonight's session is being simulcast in Spanish. If you would like to access the Spanish translation, please click the globe at the bottom of your Zoom screen and select Spanish.
La sesión de esta noche se transmitirá simultáneamente en español. Si desea escuchar la sesión en español, haga clic en el globo terráqueo en la parte inferior de la pantalla de Zoom y seleccione español.
Thank you. Okay, welcome. So this is On the Horizon question and answer, which even if you didn't attend on Wednesday, you're bound to learn something good. So I hope you enjoy it. As I stated, we do have the link to Wednesday's recording if you want to go back and see it. And this is questions and answers. So we have a few questions from Wednesday night that we're going to answer. And then please put your questions in the chat or in the question and answer panel, and we'll go through and answer them. We have about an hour. And I would like to take a moment. I would like to thank our sponsors. Without them, this wonderful educational opportunity would not be possible for us. So we appreciate them. Thank you, sponsors. And I'm going to stop sharing my screen so that we can all just chat about emerging therapies. Let me introduce our speakers. We have, I don't know if everybody sees the windows in the same order, so I'm just gonna use the order on the slide from the original presentation. We have Dr. Allison Wheeler, Dr. Robert Sidonio, Dr. David Clark, and Dr. Lisa Hensley, here to answer your questions and help you learn some cool science. So here we go. Are you guys ready for the first question?
Go ahead, shoot.
Okay, the first question is, are there any updates on the timing of Pfizer's phase 3 clinical trial of gene therapy, fidanacogene? Are we still going to get a look at initial data in 2021? Or does anyone know?
I can comment that, you know, at ISTH, and correct me if I'm wrong, Lisa, but they reported out some data from the phase 1, phase 2 data, and on surgery and liver health. And so you can actually just Google that ISTH and type in the drug. But it isn't very clear when we're actually going to hear about it. We think maybe it'll be early 2022, but it's a total guess. I'm not privy to that information, so we'll just put it that way.
Correct, and neither am I. So I think that's the best suggestion that we can offer people at this point in time. Over.
So maybe we'll see, maybe at EHOD or believe me, when it comes out, you'll see discussions. There'll be emails. I mean, they'll be available online to you know, to shareholders. You'll hear about it before, so it will be released online. At least before, so you'll have some access to it.
We've all got our ears to the ground, so as soon as it hits, I'm sure it's going to be like a little wildfire going through the community. Okay, next question is, what is on the horizon for Factor 11 replacement products? Are we behind Europe in this area, and what is being done about this?
Yeah, I can comment a little bit on this. Because I had remembered an article when we looked at this question. So there is a product, a plasma-derived Factor 11 product called Hem 11. It's a cool name for a product, to be honest. It's been around since the 90s. The problem with severe Factor 11 deficiency is it's very difficult. So plasma doesn't usually have that much Factor 11. Patients with very severe disease can develop neutralizing antibodies at very high rates. And there is a high risk of thrombosis and DIC with Factor 11 replacement in general. So I think there's been a lot of concern about bringing this product over from France to the US. And I was talking with Dr. Wheeler before. There was a brief period where they were considering it, and then the company decided against it. They were concerned about not having those issues. And there was an interesting paper, you can look it up on Blood, in which they were taking samples. These were not in patients, but it was actually specifically Benefix, and we try not to talk about the brand names, but in Benefix, there's a very, very tiny amount of 9A or activated 9 that actually is enough to stimulate thrombin generation in these patients. And they determined there's no extra 11, and it isn't the 9, but it's the 9A probably. Again, there's no clinical data on this, but it's very interesting preclinical data that there may be a role. Of course, that paper came out and I haven't seen anything happen from it, but something worth considering. Hopefully they do some kind of trial. And so we'll have to ask Pfizer. We got 2 questions to ask them about the gene therapy and about this. So
No, we should just get them on the horn right now.
yeah, they should just jump in. If you're out there, just come on and say something in the chat.
Does anyone else have a comment about the factor 11 replacement, or the next question?
It's not coming, unfortunately. It sucks, but it's not coming.
And I was just gonna comment that, given how, heterogeneous and how variable the bleeding symptoms are with factor 11, I think that there's a lot we don't understand about the protein and how much role it plays in the fibrinolytic pathway versus the hemostatic pathway versus the prothrombotic pathway. It really has its hands in everything. And so in speaking with one of my colleagues, Dave Galani, he's always commented that concentrated product just has more potential for harm than good because of the complexity of the protein and how we can't specifically target just that one hemostatic part that we'd love to be able to target, unfortunately.
Yeah, it's an interesting factor for sure. And if anybody thinks it's going to happen, Dr. Galani would be the person that would know about it. He is Dr. Factor, he's Dr. 11, so.
Yeah.
Dr. 11. Excellent. Okay, next question. Is there anything new in the works for factor 5 deficiency?
I'm not aware of anything for factor 5 deficiency. It's pretty rare. It's definitely, it's in somewhere in the middle on the rare part, rare factor bleeding disorders. And so, there certainly is a factor 10 product and 7. It's just, it's pretty rare. And the majority of the patients are fairly mildly reduced. And so, it would be very unlikely to see a specific factor 5 product.
I'm just going to ask a backup question to that. Do the adjunct treatments like tranexamic acid and stuff like that, can those help a factor 5 person?
Yeah, you want to take that, Allison, or go ahead?
Yeah, it's definitely going to be variable between individual patients, but there's potential for them to help. The other thing that can really help with factor 5 deficiency, and you can plus minus whether this is a better or a worse thing, that is platelet transfusions, because so much of factor 5 or activated factor 5 is within the platelet membranes. And so often if a platelet transfusion can be more effective than a plasma transfusion in those patients, especially if it's a non-plasma reduced platelet transfusion.
I've never done that. I remember that tidbit always comes up, but I've never done that. I don't know if you've done it, Dr. Wheeler. So
I haven't had to do it, but I've in just researching things, I've talked to people who have, but not personally, no.
Yeah.
It's another Dave-ism, and I went down.
Yeah, I'd be curious if anybody's ever done that before, because it is in the granules, right?
That's the thing about the rares, right?
Yeah.
You don't get a lot of practice.
It's tough because, there's things like factor 13 deficiency that have 2 products. And so it's tough. So oftentimes drug companies look at these products, and if there's some issues developing it or potential, problems like 11, there's usually a good reason why they haven't created it. So.
The next question is, everyone can answer, but the question is directed to Dr. Sidonio. Looking into the crystal ball, do you see gene therapy products being sufficient to control bleeds during a major surgery, such as TKR or organ transplant?
Look into my
TKR would be total knee replacement. For those
yeah.
who haven't
So my personal feeling is, I'm a little bit more excited about, not that I'm not excited about hemophilia A, but the hemophilia B gene therapy studies, they look very promising to me, particularly because there's the potential in one of them in which you could have a low titer of neutralizing antibodies, and it doesn't seem to matter that much. So that makes it more available. And the levels that they've been achieving are quite remarkable because they've been using this special variant, this particular variant called Padua. And so if anything is ever going to achieve it, it probably is the hemophilia B. I just don't see the factor VIII gene therapy ones achieving levels in the 30s, 40% consistently. So maybe if you do the surgery really quick right after it, you might be able to, but it's going to be a while. I would not bank on that for version 1.0. Check back to HFA 2031, maybe we'll talk then. So I know, Lisa, I don't know if you have if you think it's going to advance faster than that. I think it's going to be a little bit slow to get to like 50, 60, 70% levels.
No, I think that's realistic. I mean, we are seeing some amazing changes in the way that we are optimizing gene therapy and the way we're looking at expression, but it is going to be a slow process. And don't forget that regulatory process is also going to be slow.
Yeah.
So where it actually becomes available to the population, that's another hurdle. So I think your timelines are on target over.
Yeah, we'll be having HFA still for another 10 years, don't worry.
And we'll be doing this session.
Yeah.
I think what Lisa just commented on also sort of speaks to one of the questions in the chat about, in this whole conversation about people with moderate or mild hemophilia receiving therapy. I think there's always that concern of going too high, right? And but I think also with hemophilia A, it's going to be really hard to rationalize somebody having moderate or mild disease, maybe only getting 10 or 15 additional percent. Sort of the bang for your buck isn't quite as dramatic, at least from a numeric perspective. But I do wonder, and I'd be interested to hear what the other panelists think about, especially moderate patients with factor IX. I could see that coming not terribly long after patients with severe disease, just because of you're not going to get so high that you're going to be concerned about the prothrombotic component. And, we are seeing such nice success, at least with those products at this point.
Let me go ahead and read the question for those people who are watching who don't have their chat open.
Oh, sorry.
Will patients with moderate or mild hemophilia likely be able to receive gene therapy treatment? And will that decision be made by health insurance companies?
The last part is
I
yes.
can say something.
Yes.
Okay.
Yeah. So there's two parts of that question. I think it's really important to separate the two components of that. There's one where there is the regulatory approval where you've shown that a product is efficacious and is safe, and that the potential benefit to the person receiving it outweighs the potential risk. And that's a regulatory decision.
Yeah.
And then there's the separate decision about the health insurance companies. And that's a separate conversation. And it's one that, as a parent, I personally, have those concerns myself about, will the cost of gene therapy balance the cost of what prophylactic treatment is, or if you're not on prophylactic treatment, if on-demand treatment is going to cost. So I think you have to separate those two questions, which is the first one is, will there be an indication, and will it be approved, and will there be benefit? Will it be clinically indicated for patients? And I'll refer to our clinicians for them to ring in on the clinical indication. And then the second half of that will be, will the health insurance companies pay for it? Over.
Yeah, I mean, when it comes to gene therapy, we still got to get approved for severe disease. So, and maybe in hemophilia B, moderately severe disease. And so, I mean, yeah, I agree 100% with that. So, things have to be perfected in the most severe of patients. And then maybe over time, to be honest, I think we really should take a step back. We need to have factor trials for non-severe hemophilia. We've rarely had any non-severe hemophilia factor studies. Those are almost non-existent. And even though some products like emicizumab don't have a severity indication, the insurance company surely is going to have a say so in this, And so we really need to focus on that group trying to get factor therapy, because I think, there can be a lot of personalization that can be done in non-severe disease. With some patients that are on once a week, some on twice a week, and that's more than enough. Some people only do it when they're doing jet skiing and, during the summertime. And so I think there's a lot of personalization that can be done in there, but I think we need to perfect it, and we need to make sure that the risk is definitely worth it when in patients with non-severe.
Okay. Dr. Clark, do you have anything to add to that question?
No, nothing else. I think everybody pretty much got that right on the day, hit the nail right on the head.
Right on the head. Okay. What is available for someone with hemophilia B who has allergies, so anaphylaxis, to factor 9 besides Novo-7?
Yeah.
Well, there is a. Go ahead.
I was going to say Alprolix uses a human embryonic kidney cell line. If the issue is, if it's an issue about hamster cell lines, that's fine. If it's an issue of anaphylaxis because you have an inhibitor, then that's not going to work. But certainly Alprolix uses a human embryonic cell line, and the other ones use, the Chinese hamster ovary is what they're saying that show, which most products use that. And Rebenine uses that as well. And so, but I'm pretty sure Alprolix is the only human embryonic kidney one. But
Okay, so what
somebody want
you're
to check
saying is
that.
that someone could be having an anaphylactic reaction because of other ingredients in the factor, or they could be having a reaction to the factor.
Yeah, so hemophilia B inhibitors is a little bit different than hemophilia A inhibitors. You want to talk about a group that has almost no studies. There's like 1 or 2 papers that we all used from like 10 years ago on a short series of patients. There's no guidelines on hemophilia B inhibitors, but those patients, when they develop a typically severe disease, they can develop hypersensitivity. It can be something as like a scratchy throat, itchy face, or it could be full-on anaphylaxis. And and Dr. Wheeler knows as well. I think we've maybe we had a patient at Vanderbilt undergo this, but we've done desensitization, and you have to actually admit the patient to the ICU, give incrementally tiny doses of factor 9. You have to give steroids, IVIG, and following that, they typically are on CellCept as well. So it's a pretty large undertaking. We have a few patients in Atlanta that were able to successfully tolerize, but it's not for the faint of heart. If the issue is related to inhibitors, if it's a cell line, then it's certainly something to think about. Alprolix is a human embryonic cell line. I don't know if that many people have hamster allergies, but there are definitely out there.
I think Robert might be meshing 2 questions, because the next question down from the one you asked, Lori, is a woman who mentioned she's allergic to the Chinese hamster ovary cell line specifically. I don't know if that's what you were.
Oh yeah, I think I was looking at that question. I don't know if they're the same person or not. So
both questions. So I think we should also answer the question of, if you have an inhibitor, other than Novo-7, what might your options be?
Well, Dr. Wheeler can answer that. There is potential other options. Yes.
Yeah. So I put into the Q&A, but 7-FAP would be sort of the the very similar, but not exactly the same product as Novo7, another recombinant activated factor 7A, or activated factor 7, but a different molecule, and people do have different responses to 7-Fact compared to Novo7. So, so to the person who asked that question, I think that would definitely be worth, considering an option if you're not responding, or if your family member or friend or because it is not responding to the Novo7 very well. I don't know, Dr. Clark, if that's what you were going to speak to, or if there was another option that you were considering as well.
No, that was it exactly. Yeah, there is the new 7-Fact, and it is slightly different. And so from our stated knowledge, I think the only way to do it is just to try it and see how it works for you.
The only caveat is you cannot be allergic to rabbits. So it's from transgenic rabbit milk. Yes. So that's the only, I don't know how common rabbit allergies are. So, yeah, hopefully you're not allergic to rabbits.
Yeah, I do think the thing you were talking about though, Robert, is super interesting. And we did have one patient who I think was at Vanderbilt when you were there, and then came back, who was very concerned about their hamster allergy. And he had factor 8, and we tried New Week in him when it came onto the market, and he did well. So I think those different cell lines, while they don't make a difference for everybody, and don't even make a difference for everybody who has an allergy to that animal, they do to some people.
Yeah. Right.
So.
Yeah, because the Lactate and Nuvik both have human cell lines, correct? I think so.
Mm-hmm.
Yeah. Say those two again, Dr. Stonio.
Lactate and Nuvik for factor 8 deficiency. Yeah.
If you are allergic to Chinese hamster ovary, just to make sure that I heard you correctly and that we got the answer out, we should kind of mesh the questions together a little bit. Alprolix has a human line, so that one would be okay.
It's a human embryonic kidney cell line, so similar to what's used in the Lactate as well, and Nuvik. So
okay, so those are the 3 products we happen to know of right now.
Yes.
So I'm gonna skip past a couple and then back up. So be patient with me,
Sure.
audience. Thank you. I'm gonna throw this one right to Dr. Wheeler and then let everybody else comment after she's done. Is there a replacement for Stimate, desmopressin, nasal spray that you can take with you versus an IV for von Willebrand? Getting the IV DDAVP is scary because the drug loses some of its potency by the time I have surgery. The ER did not have it available when I had a serious wound/bleed.
So what I consider to be the easier answer, because we've been doing this for my patients, is subcutaneous DDAVP. If you respond to intranasal, some of the really early studies showed that you would also respond to sub-Q, and we've had patients who've done well with it. It burns a little bit, and it is a number of injections for an adult-sized person. It's usually 3 subcutaneous injections. Some people are bothered by that much more than others, and I don't judge in that regard. But that is an option. And then somebody, I can't remember who because it's not out yet, is trying to recreate the intranasal. I don't know if anyone else on the panel.
Stack Pharma, yeah.
So that's coming, but I do believe it's fewer doses than the intranasal, and it expires faster, and it's going to be not prolifically available. So I sort of a limited number to HTCs. And I'm seeing sort of follow-up questions about the sub-Q. I can look up the brand and if you type in the Q&A, I can give you the answers to that. We have not had patients. We've given the sub-Q DDAVP to probably about a dozen, maybe 2 dozen patients, and no one has complained about bleeding under the skin. But I don't know specifically for the person who asked that question if you would or not. I think if you've demonstrated that before, it might be a worry. I apologize. Our numbers are small. But I
Yeah,
can look up.
if you have very severe disease, then DDAVP is not going to be for you if you have that much bleeding. But yeah, I mean, I'm kind of curious about the IV DDAVP. I'm wondering if they're dosing it correctly, because if anything, IV DDAVP should be actually more effective. You do have to, the timing is different though, and that's where people get confused. You know, Stimate, you know, it typically takes a little bit longer. Remember, it's got to go from your nose to the blood vessels before it has its effect. The IV is very quickly in, and it should be working in 15 to 30 minutes. And the Stimate may take up to an hour to have an effect. It's similar to sub-Q. I think it's 60 to 90 minutes, right, Allison?
Yeah, the people that we've had to do trials on, we've done them the same to the intranasal, and and had similar responses. Yeah.
So the IV should be effective. There's a standard dose for it. It's mixed with normal saline, and usually it's what is it like? It's not that much. It's a small volume. We're talking about like, what is it, 30 mils or something like that. So it's a pretty small volume.
Yeah, so it's 4 micrograms per mil, and it's the same dosing as the IV. So I think we topped out at 20 micrograms, and and that was 3 injections.
So that may be what it is. You know, one of the things that we don't know about is what is the max dose. And some people use 20 to 25. What we're worried about is, you know, the higher the dosing, you start to get into a lot of side effects with no benefit. You call it tachyphylaxis, and then you end up with terrible nausea, potentially very low sodiums, and you don't get much more benefit. So IV should utilize, it actually should be better than nasal, because we, you know, in the studies I've done, it works very well. So
I think
it's just not convenient. It's certainly not convenient. And a lot of specialty pharmacies will not go out and do it. So
right. She's talking about, you know, you have to get to the hospital, you have to get to the infusion center, you know, you have to wait
exactly.
Come all the way to them. And so, you know, and then by the time, and then they might give it to you, but then by the time you're checked in and ready for surgery, quite a bit of time has gone by. So maybe they gave it to you too soon, and I can see that being
we know how long it takes to get a patient checked in. So that definitely can lose potency while we're waiting for a patient to get checked in, for sure.
Okay, so backing up, as I promised to back up, so maybe off topic, but does anyone have knowledge about a knee replacement in a naturally fused knee?
Do we have the disorder? Or
I'm assuming that we're talking about a knee that was fused, you know, from hemarthrosis and damage and whatnot. Maybe I'm wrong though. So.
Yeah, I mean, I think that's where you really have to rely on our orthopedic surgeons, to be honest, about the benefit. I mean, they factor in a lot of things. Do you have an appropriate therapy that's going to control bleeding afterwards? Is the patient going to be reliable and do prophylaxis if that's what's needed? And you know, obviously they have to be old enough to make it worthwhile, because you know, in a young patient, it may not be as beneficial. So really you have to rely on our orthopedic colleagues on that one. That's where we defer to them on those decisions. So sorry, I don't know if that's a strong answer for that. So.
Okay, I'm looking for more questions, and while I wait, I'm gonna ask a question. Does anyone feel like they want to describe the difference between, or the similarities, differences, whatever, between the mRNA products being tested for potential bleeding disorder support versus these mRNA vaccines we're all hearing about?
Lisa or David, do you want to take that, or?
Actually, I am not. There is a company that was trying to develop an mRNA for factor 9, and I haven't really heard much from them for a couple of years, so I don't know how they're doing there. But it's basically the same idea, is that with the COVID vaccine, the mRNA gets in the cell and it causes the cell to make the COVID spike protein. With the factor IX product, it would be the same thing, that the mRNA gets into the cell and causes the cell to make factor IX. Now, I don't see the benefit of that actually over just giving somebody straight out factor IX, but there may be. But yes, there is a company that was working on that. I don't know whether they still are.
Yeah.
Hi, I can add a little more, Dr. Clark, to that. So there's a couple of I think it's important that we don't get confused between the mRNA vaccines and what we refer to as the RNAi or the siRNA.
Yeah.
And these are two very different things. And I know that's not what you were talking about, Dr. Clark, but I want to make sure for the folks listening that we make that distinction. So RNAi, or what we call small interfering RNAs, is a strategy where we actually use the body's natural defense mechanism. So we, as humans, don't normally see double-stranded RNA. If our body saw double-stranded RNA, it would say, we have a foreign invader. That's likely a viral infection. I need to get rid of that. So the scientific community, probably about 20 years ago, recognized this, and about 10 or 15 years ago said, hey, maybe we could start using this as a therapeutic. And they started making short pieces of RNA that would bind to messenger RNA in your cell. And when your body saw that, it would actually chop that up. And it was a way to downregulate or to develop potential therapeutics. And so we started using this for a number of different indications. And I'll let Robert talk about the drugs that are in development. The difference between what we're doing for COVID and the vaccines is we're actually opposite direction. As Dr. Clark said, we're actually delivering the sequence of the virus and packaging it so that our virus says, hey, I should make this protein and I should express this protein on the outside of the cells. And the benefit of doing that is that it looks like my cell was infected by the virus. So my body develops an immune response that is very much like the immune response as if I'd had the infection, but without the risk of the infection. Now, where these technologies come together and where there's synergy between these technologies is the trick for all of this. And some of you heard me talk about gene therapy, but where the trick is for whether it be an RNAi or an mRNA vaccine, any of these is how to get the RNA to the cell. And our body's RNAs traditionally are not very stable, which is why many of you have heard challenges with the Pfizer vaccine or the Moderna vaccine where it has to be stored at certain temperatures. But we've heard that RNAs are not very stable. And so we have to package them so that they're protected, they don't get destroyed, and that they actually get into the right cells. So the same technology that a company named Alnylam, which Robert may mention, and another company, Techmera, developed years ago, these small lipid nanoparticles, are actually the same technology that's being used to package our vaccines. So there is where these technologies merge is how we deliver them, but they are very different technologies. And Robert, I don't know if you want to mention a little bit about the drug and how that drug is actually working, and jump in here over.
Yeah, and David, I don't know if you want to. I know you talked about fitusiran in your part of the presentation, I think, right? Yeah, sorry. Go ahead. I'll let you speak, and then I'll jump in and talk a little bit about some of the delivery stuff. Yeah.
Yeah, one of the things I was talking about during the regular session was rebalancing agents, where we're trying to, because you have hemophilia and you don't have factor VIII or factor IX, the clotting system is out of balance, where normally it would clot when it needed to and not clot when it shouldn't. But because you're missing a major clotting factor, your clotting system doesn't clot well. So one of the ideas people are working on a lot is trying to reduce the amount of anticoagulants. That's part of the control system in the clotting cascades. Some people call it the brakes on the system, but it really keeps the clotting under control. But if you don't have enough factor VIII or factor IX, it's really providing too much of a barrier to clotting. So we're trying to, some people are trying to reduce the numbers of the amount of anticoagulant in there that's holding back the clotting process. So like Dr. Hensley was talking about, one of the ones that seems actually like it may really turn out to be a good product is fitusiran. And this is one where they're using the interfering RNA. So they're using this interfering RNA to actually stop the cells from producing antithrombin, which is one of the major anticoagulants. And so by reducing the amount of antithrombin, they're better able to restore the balance in the clotting system, and so that the system actually performs much better, more like it's supposed to, even though you don't have factor 8 or factor 9.
Yeah, and the great thing about these, these small interfering RNAs is it's unlikely, one is they're relatively inexpensive to make, and two, it's unlikely that there's going to be a problem. There hasn't been an issue where it's not going to work. Your body isn't going to create anything to clear it. And they are biodegradable, if you can use that term. They will disappear. And but the problem is, it's these targets like antithrombin and targets like protein S. It is possible to target them, but there's like a fine line. Like if you cross that line, you can get into a lot of trouble. And I feel like it's tough in different patients that may have different. We may have to screen patients for other, common inherited clotting disorders like factor V Leiden mutation. It's about 5% of Caucasians have it, so it's certainly very common. And I don't know if we're going to have to screen for that before we consider therapies like that. Going back to what Dr. Hensley was saying, there's those lipid nanoparticles. They had attempted years ago. It's been 4 or 5 years ago now, I think, David, when they talked about delivery of the mRNA to sort of target it and make these mice make factor 9. It's been a while. I haven't seen anything else come out from it, so I'm not sure if it was just too technically difficult. An interesting one I don't think we talked about is that Sigilon Therapeutics has attempted to have these engineered cells in which are programmed. They're sort of created in this like bio matrix to prevent fibrosis. And it actually kind of looks like, if you've ever had bubble tea, when you look at it, it kind of looks like bubble tea. Not anything you want to ingest by mouth, but unfortunately you have to inject it into the abdomen. So it requires minor surgery. But unfortunately, and the idea seemed great, that you could inject these little beads that have those engineered cells, and they could make factor 8, and you might be able to have mild hemophilia for potentially a decade or so. The problem is once the dosing started going up, they had to put a hold because there was a development of a patient with an inhibitor, and these are previously treated patients. So there is currently a hold on it. I don't think it's changed, but it's I'm pretty sure there's still a clinical hold. I don't know if it's been released. It was as of July. So we'll see. The problem, the thing that we didn't really talk about is that when you take these products like VWF, factor 8 or factor 9 in its sort of wild type normal state, can be very difficult to inject it into the body and not have the body try to create antibodies to it. It gets exposed to the lymphatics. That's probably what happened with Sigilon. It probably had to go through the lymph, and that exposed it, and unfortunately developed antibodies. And so VWF doesn't seem like you could inject it. Factor 8 certainly does not want to be injected into the skin. But we'll see. We talked about the factor 9 sub-Q product. We'll see. They so far have had minimal inhibitor responses. They had a set of cousins develop it. But we'll have to see. Hopefully they're the only ones that will develop it. So it's tough. Yeah. We want to make sure we emphasize with patients, it's not for lack of trying. You don't see all of the drugs that fail. They might get to mice and then they never make it past that. And, you don't see all the failures, the expensive failures that happen before one of these breakthrough products come through. I was looking back at emicizumab. They first started working on that in 2002. It took a freaking decade before they could get that product into humans. And so, you know, it does take a long time, unfortunately.
Yeah, and actually another thing I was just thinking about when I was talking about the rebalancing agents where we're tweaking the clotting system.
Yeah.
We started out, sort of bemoaning the fact that there weren't concentrates for things like factor 11 and factor 5. But if these rebalancing agents work out, actually that may be a treatment for almost any type of a bleeding disorder where you're missing one of the clotting factors, that you could potentially kind of tweak the clotting system to clot anyway, even without the missing factors. The big problem in these things is that you're kind of like it is a balancing act, that you're trying to not cause so much clotting that you end up with thrombosis, which would be a big problem. And so I think actually my idea with these rebalancing agents is they're going to need to perfect some kind of a good clotting assay that kind of shows the overall clotability of your blood, so that you can kind of adjust it the right way. I mean, now when somebody has a breakthrough bleed with one of these rebalancing agents, there have been several cases where they've given them, extra factor, or for people with inhibitors, they've given them bypassing agents, and they've been, pushed. The balance has been pushed over too far, and they've experienced thrombosis. So if we had a good way to kind of just look at the blood and see what its overall clotability is. And one of the assays that people look at is a thrombin generation assay, but that's a very tricky assay. And no two laboratories get the same results with that. So, but I think if there were a need that people could potentially work on this and come up with some way to do this.
Yeah, the protein S is another target that people are working on right now. But go ahead, Dr. Wheeler.
Oh, no, I was just going to say, I echo Dr. Clark's comments about, how, or what he implied, and that things are going to get more complex. I tell the fellows that I work with now that, the different dosing between factor 8 and factor 9 is the easiest thing they're going to have to deal with if and when these products come to market, because it's not just going to be about understanding the factors and understanding what these patients are getting, but also knowing that the mistreatment, could have truly deleterious effects. And so hemophilia is so exciting right now from a research and a development perspective, but I think we're all going to have to take a deep breath. And I have noticed a couple of comments in the chat about the frustration about, the ERs not having products. And I think we all deal with, patients traveling and going to places with small hospitals and us begging them to call us so that we can help guide their treatment if they do have a bleed. And I think we're in for it in that regard, because while these products have a lot of potential, the potential's on both sides, and we're going to have to be careful. But it doesn't mean it's not exciting. I'm super excited about it. I just also know that there's going to be a high learning curve for all of us.
They're trying to go after these targets. Protein C seems like a potentially scary one. Protein S may be okay. Antithrombin is pretty close as a target. It's actually pretty close to the finish line here. So we'll have to see in 2022. We'll all keep our fingers crossed. I'm not privy to any information, but I'm hopeful that we see that submission soon. So if it hasn't been submitted already.
It sounds like it would be a little riskier to play around with those off-label if they get approved. It's not like, well, if your factor level is low, maybe we'll just try giving you a little factor because it really cause a thrombotic event.
Yeah, they're a little harder to dial in the exact dosing. It will make it a little tougher.
Yeah, but still very exciting. Because some of those bleeding disorders out there that kind of have nothing might get a subcutaneous solution that works for some time.
Yeah.
So,
or alluding to the comment or the question from earlier about patients with hemophilia B with inhibitors and allergy, If these products come to fruition, they will be added to that list, which is all very short at this point in time.
Yeah. And Dr. Hensley has put in the chat, I hope everyone can see it, if you're curious about the delivery system for RNAi and RNA vaccines, there's a Forbes article from August 17th of this year. It's called COVID's Forgotten Hero. So those of you who are asking questions about that, I think I started that question, but I know a few people were thinking it. So there's that information for you. And yeah, as far as, I'm just gonna make a comment about hospitals being unprepared. This is a common occurrence. And my advice as a patient, and it's not fair, and yes, they should be, but we're rare. And some of our medications don't have a very long shelf life. And so especially smaller hospitals, but even larger hospitals might not have it on hand. Especially if someone went through the ER, 2 days before you with a problem, they may have used up their supply on that. So the smart thing is to try to have products with you. And anytime you travel, please notify your HTC that you're going, because a lot of times they can put you in contact with the HTC where you're going to end up, and you can know how to find them and who to ask for and all of that kind of stuff. And I would just recommend, it's on us, and it's not fair, and it stinks. But
if it's an academic hospital, they're most likely going to have some things. Sometimes we have to have things on consignment, so we don't eat the entire cost if it expires. Some of these products are like $10,000. It's hard for a place to just eat that cost, watch it expire. So if it's those, but things like ddAVP and Amicar or tranexamic acid, those are relatively inexpensive and should be had at most hospitals. It's really important to just talk to them. We've had some hospitals where we've negotiated where there's patients nearby, and so they'll stock it. If you have that conversation with them about it, if they know there's 1 or 2 patients with it nearby, they will stock it. But if it's a small rural hospital, those places barely are staying afloat. They can't, it's just hard for them to hold expensive factor products.
We carry it with us. Sometimes I have a whole suitcase. Okay, we're just pretty much at time. So I'm gonna go ahead and wrap it up. I want to thank my panelists. You guys were rock stars. You're awesome. This has been a wonderful conversation. I have really enjoyed it. I know that the audience has as well. And then I will, for my, formal, I don't know what to call it, formal ending of the session, I will share my screen and I will say thank you to everyone. And then really quick, I will also go back to our slide from last night where we had some resources for you. These are the 3 primary websites I recommend. Obviously, once you get into one, you're going to rabbit hole all over the place. But know that we're here for you. And if you need advice, please do reach out. And if you need someone to talk to, please do reach out. And let's keep the conversation going about emerging therapies. There's a lot of hope out there and a lot of fear, I know, but a lot of hope. And so thank you all for, the panel worked very hard preparing for this. So thank you all for your hard work and your brilliant minds and for sharing them with us.
Have a good night.
Pleasure to be here. And thanks for everyone else for joining us. Over.
Thank you all for joining us.